claim that one individual cell will go through this cycle repeatedly. It may stop at certain stages, exit the stage, and never continue with the cycle. Okay, so first and foremost, we recap that there was G1 phase, Gap1 phase, a lot of growth and takes place over there. Then there's synthesis. We know now that this synthesis refers to DNA replication. What is expected of you for this section? You don't need to know the mechanism, but you have to have a deep appreciation of the experiment. Next, it goes into G2 phase, where more growth takes place. I mean, makes sense, right? We need to have more cytoplasm, more enzymes, more proteins before we split the cell. If not, every division we go through, you have less and less things. Then we go into PMAT, prophase, metaphase, anaphase, telophase, and finally cytokinesis, and then you get two cells. Each of those two cells then go through their own cell cycle after. Okay? I want to talk a little bit about what the DNA looks like at each phase. First and foremost, at G1 phase, let's assume we work with yesterday's numbers. This particular cell has 6 chromosomes. In other words, 3 pairs. What would these 6 chromosomes look like at G1 phase? Likely, they will be present inside the nucleus. And the thing is, these 6 chromosomes, they are unwound. Okay? So 1, 2, 3, 4, 5, 6. It's a mess. You often cannot tell what it looks like under the microscope. They just look like dots. What will it look like in after S phase? So let's say after S phase is gone through DNA replication. And if we were to look into the nucleus, what would it look like then? Actually, it would look kind of like this messy state again. But if we were to look very carefully, I'm going to draw, okay, it looks very messy. But in actual fact, let's pull out one of them. It will look something like that. Okay, so one like that. But after DNA replication, I should have one more DNA molecule, but attached at this region called the centromere. There should be 6 types of this structure. Following? If I were to zoom into one of these DNA molecules, you should find 2 DNA strands. Following? Maybe I'm not clear. I felt that I was not clear, so I'm going to redo it. If I were to take one of these DNA strands out, this DNA molecule out, if you were to look very carefully, actually it consists of double strand. Yeah, because all DNA is double stranded. Okay, so you have 6 kinds of this. After DNA replication, what you should see is double stranded DNA, but because we've just replicated the DNA molecule, you should have one more molecule that's double stranded attached at the centromere. And I should have 6 kinds of this. Yeah. So, okay, messy state, but overall times 6. Okay, so a quick test. If I were to just play with words now, how many strands of DNA is in this particular nucleus? How many strands of DNA are in this nucleus? Okay, so listen to the terminology. If someone were to say DNA molecule, often they are referring to 2 strands together. Okay? If someone were to say DNA strand, they are referring to just one half. Okay? If someone were to say chromosomes, chromosomes are referring to DNA molecule. Each molecule is made up of 2 strands. Following? So at this point over here, how many chromosomes do I have? I have 6 chromosomes. 6 DNA molecules, in other words, 12 strands in total. But after DNA replication, how many DNA molecules do I have? It's interesting. Let's go with all the terminologies then. How many chromosomes do I have? How many DNA molecules do I have? And how many DNA strands do I have? See, how each term refers slightly different things now. I have still 6 chromosomes. Yeah? Because remember, we refer to one entity as a chromosome. How many DNA molecules do I have? If I really count up, there's actually 12. Although 2 DNA molecules are always attached to each other. Finding how many strands, which I have 24. Okay, I'll never test you this in the exams, but just appreciate. Appreciate so that when you write it out in a sentence, you don't use them interchangeably because they do mean different things. Because if you were to tell a teacher that there are 12 DNA strands over here, it is actually factually not correct. It's not even a spelling error, factually it's not correct. Okay? Let's progress through the phases a little bit more. Okay. At prophase is where we know the DNA molecules start to condense into more compact form, easier to segregate. So what would the nucleus, or rather no more nucleus at this point, the nucleus will have broken down. But each one of these structures, what will it look like by this point? It will look something like that. Small little butterfly structures. Yeah? Every chromosome, this is a decondensed form, will now become a very condensed form. Okay? And we refer to this particular structure as a metaphase chromosome because you only see this from metaphase onwards. Yeah, pause here for a while. This chapter has tons of vocabulary, new ones, yeah? Then the worst part is that they are very similar sounding. If you are good and ready, look at the whiteboard. I have 1, 2, 3, 4, 5, 6 chromosomes there. If a teacher were to show you these 6 chromosomes back on the whiteboard, and they give you an MCQ question, they ask you which phase do you think this cell is most likely at? 4 options: S phase, G2, prophase, metaphase.
Is there a nucleus?
Okay, is there a nucleus? Good question. No, I did not put a nucleus there. Which phase do you think this cell is at? S phase, G2, prophase, or metaphase? Hey, sorry, I never even organized this properly. If I organize it, it's too obvious. Alright, okay, we know the answer. We cannot. Sorry, that was a stupid question. But I want to reiterate, you should only see these structures at the end of prophase in metaphase onwards. Okay? You shouldn't see these structures at the end of S phase. Yeah? These butterfly structures only exist after the DNA is coiled very compactly. In other words, if I were to look at this structure over here, inside here you have the DNA tightly wound. Very, very compact. If I rewind the clock, I can actually untangle all of this and make this much, much longer. Okay? And mind you, this is all double-stranded. Okay? Some additional terminology you may have encountered. What do you call just one half of a metaphase chromosome? Is sister chromatid. Okay, so this is a vocabulary you need to be familiar with. Something that often comes up is that people tend to say that sister chromatids are genetically identical. But I wonder, when you saw this, whether it makes sense to you. Sister chromatids are identical to each other, genetically identical to each other. Does this make sense? And if someone were to ask you, why is this identical to this? Well, actually the response would be, because of DNA replication. When you replicate DNA, the intent is to create an exact copy. So sisters are actually identical to each other. In fact, I would dare say therefore that these sisters are twins. Okay? They are identical DNA molecules, just attached together at the centromere. Yes, Valentina? Oh, as in, can I still call it a sister chromatid if it's uncoiled? You can. I've not seen people, we don't actually have a term for what's the uncoiled version, but can call sister chromatid, because we have no other term for it. Okay. So Valentina is asking, compact version, 1/2 sister chromatid. But if I were to uncoil everything, okay, so if I were to uncoil everything, one of these long uncoiled DNA molecule, do I still refer it as sister chromatid also can? They are sisters because they are attached to each other at the centromere. And they are genetically identical because they were, if all went well, if replication went well, they are identical. One last term I'd like to introduce to you that you may have encountered. Homologous chromosomes. If you want to refer to your notes, I believe that you can find it on page 10. Page 9 and page 10. Homologous chromosomes. I'm going to spend a little bit of time talking about this term. Hopefully you can appreciate it better. And then you feel more confident to use it when you write. What are homologous chromosomes? say that this particular cell has 6 chromosomes, 3 pairs. You can see 3 pairs here. Yeah. Why don't chromosomes just pair up like that? What makes them pair up in this way? Okay, if we were to look at each of these pairs, the reason why the cell organizes your chromosomes this way into these pairs is because they share very similar structure and property. In other words, if I want to elaborate what the features of homologous chromosomes are, number one, they have the same length. Okay, lengthwise, they're the same. They also have the same genes. What do I mean by same genes? You have a gene for everything. You have a gene for skin color, a gene for hair color, eye color. So I'll give you an example. Let's say this particular chromosome at this point over here, I have the gene for eye color. Both of these chromosomes at this same exact location on the DNA can find the gene for eye color. Maybe if we look at another chromosome, we may find at this point over here, we have the gene for maybe skin color. What you won't find is this gene. What makes the homologous chromosomes the same pair is if they share the same length and the same kinds of genes present. Would there be other genes present? Yeah, maybe at the very end over here, maybe you may find another gene. Maybe it's a gene for what other things do we have? Yeah, I'm just focusing in color because it's easier to visualize. Maybe for hair color. Okay, that's what makes homologous chromosomes homologous. And therefore, the origin of the name. Homo means same. Same chromosomes. Homologous chromosomes. Humans, we have 46 chromosomes, 23 pairs of homologous chromosomes. Yes, Zoe?
This is during phase.
Oh, at what stage is this at? Okay. These genes are present at any point in time. If I rewind the clock, let's say I'm looking at this particular DNA. Maybe at this point at G1, this DNA still exists but uncoiled. Somewhere over here is the gene for eye color. Somewhere down here is the gene for hair color. So these genes are always present at any time of the cell cycle. Okay, the reason why I'm bringing this particular term up is because sometimes they pair up, sometimes they don't pair up. Okay, and for those of you that really studied meiosis, you know that in meiosis they pair up. Okay, but they don't anyhow pair up. They must pair same chromosomes. I'd like to make a slight correction. Sometimes people consider and say that we only have 22 pairs of homologous chromosomes. Sometimes because actually the final pair of chromosomes within all of your cells, okay, the last pair, the two chromosomes aren't exactly the same. Those are your sex chromosomes. Because the X chromosome is much longer than the Y chromosome. We do not consider this homologous. To be homologous, you need to be same length, same kinds of genes present. So the last pair of human chromosomes, sometimes we don't consider it homologous. But some sources just anyhow whack and say 23 pairs. But if you want to be very precise, this is the truth. Yes, Yihui. Okay, Yihui asks, when do you say chromatid? We use the word chromatid only when we want to refer to just one half of the chromosome. Okay? Whether uncoiled or coiled, you can use the word chromatid. Okay, I'll pause here for a while. Yes, go. Yeah. Oh, okay. You only use the word chromatid to describe one half attached to another half. The moment it's separate, the term doesn't exist anymore. It becomes two daughter chromosomes after it's separate. Isabel? Oh, 46 is the number of chromosomes we have. We have 23 pairs. Each one of these pairs is just one type of chromosome. Correct. And but in reality, we have 46 chromosomes, but 23 of them are just the same type. Sorry, wrong way of saying it. We have 46 chromosomes, or in other words, we have 23 of these pairs. Here I'm only showing you 3 pairs, but imagine in humans we have 23 pairs of chromosomes. 23 pairs of chromosomes equals to 46 chromosomes. Yes. Oh, okay. So are you saying, when I say humans have 46 chromosomes, are you thinking that yourself, you only have 46 chromosomes? Okay. Then maybe I need to qualify that better. Every single one of your cell has 46 chromosomes. From the cell in your eyeball, the cell in your skin, the cell in your toe, every cell has 46 chromosomes. Okay? You may be thinking, hey, if the cell in my head, the cell at my toe also has a gene for eye color, doesn't make any sense. How come I don't see an eyeball on my toe? The gene for skin color, this particular set of chromosomes are also present at my liver cell. How come my liver cell not like got skin color? Or maybe we deal with hair color. Gene for hair color, you also find this chromosome on your liver. How come our liver is not hairy? Okay? The answer to that is our body is able to turn on and switch off genes. Okay? So, for example, if I have a cell that is present at the eyeball, what you'll find is that it will switch off all the genes not related to the eye. For example, I will switch off the gene for hair, I will switch off the gene for skin, and I'll only express on the surface any gene that's related to the eye. That's how we are able to differentiate all our cells for different purposes. Yes?
What about different eye, different color?
Different eye, different color. That's because each eye, they are switching on and off slightly different things. So likely one of the eye could be switching off a certain color, and the other one is switched on. And sometimes you see different traits being expressed on the surface. Oh, okay. So when you say every human has 46 chromosomes, do you mean to say that you only have 46 chromosomes? Okay, so you understand, right? That you have billions of cells, every cell has 46. So if you really want to do the math, you have 46 chromosomes times how many billions of cells do you have in the body, right? Okay, so your question is?
But not everyone has the same type of...
Okay. You see, we talk about this, right? Then you realize something is not right. If everyone has these genes, right, how come we don't all look like the same thing? Right? You look different from me, You look different from me. Our hair color different, our skin color different, our eye color different. How is this possible when we have the same genes? It cannot be that you switch off some things and switch on some things, right? And the eye, that's why your color is different, mine is different. Okay. And this is why I wanted to spend a little bit of time here, because the next chapter we're going to go and move from repro to inheritance. You will see how this case makes a little bit more sense. You know that for each gene, you can have slightly different versions. So for example, version 1 could be blue. Version 2 could be black. Version 3 could be brown. So, let's say maybe we look at Isabel. Let's say Isabel, on the surface, she has brown eye color. If we go down to the cells of her eye, actually what you may see is this. She may have the gene for eye color, but she has the third type of it, the third version of it. So maybe her version is brown, maybe brown, 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 and brown, and brown. But maybe for myself, if you were to take some of my cells out and you go and analyze it up, I may have a different version of the gene for eye color. My version may be black, 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 and black. This helps explain why, even though we have the same gene for eye color, why we could have, we see different things on the surface. Because we may have different versions of the same gene. Everyone can produce a color for the eye, but we may produce different versions of it. What gets interesting in this particular topic is when some people have two different versions at the same time, and it's possible. So there are people that, for example, one of the homologous chromosomes shows black. Then the other homologous chromosome shows brown. This is possible. But what will show on the surface? Yeah. So that is the fun part about this chapter. In this chapter, we will learn about which one will dominate over the other. So to show on the surface. Usually there is one of versions that is stronger than the other, more dominant than the other. For example, brown could be the more dominant one, and on the surface, even though you have black underneath, you will show brown eye color. Okay, but that will be for the next chapter onwards. Yes, yes.
So in meiosis 2, right, it says that the chromosome is separate from daughter chromosome. So, like, does the number of chromosomes 46 remain the same throughout the entire
because after you separate the chromosomes, so will the number of chromosomes be the same with it? If you understand the definition of a chromosome, and we're still within a cell, you're talking about meiosis 2, right?
Yeah.
Okay, pause there. Everyone roughly understand the idea of a homologous chromosome? Same, but could be slightly different. Okay.
Wait, but if you're saying different, then you're not genetically identical, right?
Correct. But you see, I never said genetically identical. I said they have the same genes, but they can have different versions of the same gene. Yeah? Okay? You have the same gene, but you have different versions of the same gene. Okay? You ready? Okay, then now let's talk about meiosis. Then you can start counting already. Yeah? If you appreciate all this, meiosis has a little bit more value to you. Yeah, just ask the question. Yeah, what? Yeah, I'm going to start meiosis though.
Oh, wait, okay. So,
Yeah.
when you say humans have 46 chromosomes, you mean like that squiggly not formed together one, or like method-based chromosomes, or like condensed chromosomes, or like what's one?
here, so when we say a human has 46 chromosomes,
Yes.
at which stage am I defining
Yes.
this? Right? Okay, we usually define it at the interphase stage. Because that is when most cells are just resting, not doing anything. You may want to argue, therefore, from here to here after they replicate, shouldn't I have more chromosomes? Actually, you don't. Because it is two DNA molecules attached to one, it's still the same number. Yeah? So although I have 46 DNA molecules, double-stranded, after DNA replication, they are joined together, they still consider one chromosome. So you still have 46 chromosomes. Okay, right now we're going to go through quickly prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. Okay, ready? Okay, let's say we are at prophase 1 right now. At prophase 1, you'll find that the chromosomes are kind of just scattered everywhere. If you read your textbook, it says that something interesting happens at prophase 1 for meiosis. And what is that special thing that happens? For the first time, you will observe that if they are homologous, they will pair up. Okay, this is in prophase 1. Homologous chromosomes will pair up for the first time ever. What happens during metaphase 1? Metaphase 1, these homologous chromosomes, these pairs, will then align themselves at the equator of the cell. Correct? Yes. Oh, where does it? Okay, the moment they already join together in the pair. So metaphase 1. Following? Anaphase 1. I want you to spot as many differences as you can when you compare meiosis versus mitosis. Okay, we compare the two up. Later on, we will summarize all the differences. Anaphase 1, we will separate the homologous chromosomes. Telophase 1, these chromosomes will likely uncoil and reform the nucleus. Likely cytokinesis will occur. Then you'll have two separate cells from now onwards. Two separate cells. Let's go into prophase 2. In prophase 2, the nucleus will break down. And these chromosomes don't have a particular arrangement, kind of anywhere. We go into metaphase 2. These chromosomes will then align themselves at the equator. In anaphase 2, these chromosomes, these sister chromatids will be pulled apart to form daughter chromosomes. In telophase 2, these daughter chromosomes will uncoil and likely new nucleus will form. And after cytokinesis, I will have 4 cells in total now. Following? Okay, yes. Okay. So, understand that in a cell, whether one is on top or below, actually, biologically, it makes no difference. Yeah? It can be randomly arranged. So, there's no significance. differences you spot between meiosis and mitosis? If we practice the terminologies again, if we look at hello phase 2, how many chromosomes do you see here now? Okay, only 3 now, right? 3, 3, 3, 3. Notice that this is now half of what the original number is. Yeah, that's a significant thing you see in meiosis. In meiosis, the end result, the cells will end up having half the number of chromosomes as the original parent cell. This is important. If the sperm and egg has exactly the same number of chromosomes as the original, then every time a sperm and egg comes together, you will have twice the number of chromosomes. Then this baby, next time go and have another partner, come together, the next offspring will have even more chromosomes. The number of chromosomes will just increase exponentially every generation. So it makes sense that the sperm and the egg needs to start off with half number. So when you come together, then you will have a baby that is the original number. Okay, we compare meiosis 1, meiosis 2. Actually, between meiosis 1 and meiosis 2, which meiosis is very similar to mitosis? Okay, meiosis 1, meiosis 2. Which one is very similar in the processes to mitosis? Number 2, right? Actually, number 2 is very similar to mitosis. Yeah? It is meiosis 1 that is significantly different from mitosis. Lots of differences that you can compare at every single stage. Yes. Cannot already. Yeah, so Nicole asked a good question. After meiosis 2, could this cell go on to maybe divide by meiosis again, is it? Okay. Could it divide one more time? I don't observe this happening in biology. After you get half already, right, we will not half it one more time. There's not much value in that. Yes.
So if after mitosis, one cell divides into 2, right?
Yeah.
And then meiosis 1 and 2, one cell divides into 4.
Yeah.
So the whole process is 1 to 8.
Whole process is?
Like one cell to each cell.
Oh, wait. So you say again, let's say we have one cell, and it divides into 2 cells via mitosis, and you are asking, what if each one of them go through meiosis?
Yeah.
Oh, okay.
I thought it's a continuous process.
What is a continuous
I
process?
thought like it's always mitosis and then meiosis.
Oh,
It's 2 different ones.
oh, so there are 2 distinct processes. Mitosis is mitosis.
Oh.
Only certain cells in your body can carry out meiosis. But mind you, your eyeball won't suddenly produce 1 egg or 1 sperm. Okay? Only the cells inside the testicles and the ovaries can carry out meiosis to produce sperms and eggs. Every other part of the body should not be able to carry out meiosis. It will be very odd. Shanice, okay? Give you a yes. Oh, okay. Can you ask, does meiosis occur on any body cell, or does it occur from specialized cells? Okay, what is the parent cell for meiosis? In the context of humans, if you go down to the ovaries and the testicles, there are specialized cells there that can carry out meiosis that will form sperms and eggs. Yeah. You only find it at the testicle and ovaries. No other cell in your body should be able to carry out meiosis. Because the only function of meiosis is to produce sperm and eggs. Mitosis, any other part of the body should be able to carry out because you need to repair yourself, you need to grow. Yes, Zoe?
When has crossing over like occurred? Because it just says at the beginning of meiosis.
Okay, let's rewind the clock then. We go back to, and then my question is, at what point do homologous chromosomes pair up? When do they pair up? Okay, yeah, they pair up at prophase, right? So at prophase, they pair up. So actually at prophase, it can start crossing over already. Yeah, crossing over occurs the moment they start pairing up. Shani, did I lose you? Okay, we will stop here today. Yes, more questions for me. Yes. Oh. Now you ask the question, what in the world is independent assortment of chromosomes? Okay, so can you, earlier on I said that each homologous pair of chromosomes have same genes, right? I'm going to represent same gene with the same alphabets. Okay, so for example, alphabet A. Okay? They are identical because DNA replication. Over here, maybe it's a different version of the same gene. I'm going to use same alphabet, but maybe small caps. Okay? Over here, E is a different gene. Different version of the same gene. Yeah, okay, maybe over here, I have D, here's small d. Okay? Okay, sometimes you ask when the word is independent assortment of chromosomes. The independent assortment of chromosomes is this. You know when they line up at the equator? How they line up determines whether they go to the cell on the left or the cell on the right, correct? But how this pair arranges itself does not depend on the pair above. Okay? For example, this pair could have arranged itself this way. If you arrange itself this way, the cell on the left will have a different combination already, right? Cell on the left may have big E, small b, small a. Cell on the right will have small d, big B, and big A. Combination number one. But if this pair over here decides to pair up in a different way, I generate another combination. Big D, small b, big A. Small d, big B, small a. That's what independent assortment means. It means that every pair has its own right to decide how they want to arrange. But depending on how they arrange, the downstream effect is that they will go into different cells at the end of the day. But this is important. This allows every single sperm that is produced to have a slightly different combination. If not, you and your siblings will look exactly like the same thing, on the same factory mode. Yeah.
So this is like, you cannot control it, just happens itself.
Yeah, you cannot control. So every sperm and egg that you produce, here I only show you 3 pairs by the way. you have 23 pairs. Can you do the math? How many 23 to the power of n is it? No, sorry, how to do this? Was it 2 to the power of 23? How many combinations could I have if there are 23 pairs and each pair could arrange themselves in a different way along the equator? How many combinations of sperms and eggs will you end up with? I don't know how to do the math. But that means to say almost every single sperm or egg that we produce will be very, very, very different. They have different combinations because of this independent assortment thing.
What about twins?
Oh, twins. Okay, what about twins? Okay, twins, right? Twins form at a very late stage. Twins form after sperm and egg has fused with each other. So here I have one sperm, here I have one egg. Let's say they fuse with each other, then you produce baby. You produce a single cell first. This single cell, in the next chapter you learn it has a name, it's called a zygote. Okay. So when do twins form? Twins form at this particular stage. Before this cell goes on to develop into a baby, it splits into two via mitosis. Each one of these develops into a baby. That's why, right? This baby has exactly the same chromosomes from the original cell.
Wait, that's every zygote go through mitosis.
Every zygote does go through mitosis to form a baby. But some zygotes, after they go through the first mitosis, each one of the resultant cells, right, goes on to form a baby of its own. That's when you could get identical twins. But for you and I, that single cell divided into two, those two become part of your body, these two, and then it goes on to divide until you become one single baby. Yes? What do you mean what happens? Oh, okay. Okay, so you ask what happens if you absorb your baby twin? No, it's possible. Okay, you do not really absorb. What happens is this, here, if you have a zygote, so one sperm, one egg fuse together to form a zygote, right? Z. By right, if you want to form a twin, you will have to divide into two, each one divide into baby, right? Okay. But sometimes this division process doesn't occur very well, and you may divide halfway. Then from then on, it then goes on to develop two babies that are not fully separated, still joined. But you're asking what happens if you absorb. I mean, it happens, if you absorb, means you absorb, I don't know. What happens if you absorb? That individual that has absorbed it will never know that he had a twin. Okay? So those Chinese twins never divide properly. After that, each one of these will divide some more. Then you form yes.
Is it by chance completely, or is there a gene that makes it more?
Say again? Is it by chance?
By chance completely, or is there something that makes it more?
Say again?
Is it by chance, or is there a gene that makes it more prone?
To form twins?
Yeah.
Oh, I don't know. I have no idea. But I suspect it is related to the genes you have, because I had a friend, the mother, right, both times she conceived, right, twins. So she only wanted 2 kids, and now I got 4. Yeah? So twins, second round, kids. Yeah. Okay, I will stop here. I am over teaching some parts. Okay? But when we go to reproduction and inheritance, there is a lot more value there. Right now, you just need to understand one cell become 4 or one cell become 2. Okay? Alright. On Friday, when I take over Miss Lee's lesson, then we will start exploring the parts of your body. Okay? Male and female.